This meeting is recorded, and I think you'll see that notification pop up now. And you are muted currently. And if you'd like to ask a question, just please raise your hand and we will unmute you. Alrighty, and so for today we have a 15-minute talk from our wonderful trainee Laura, followed by a PI speaker, Dr. Mike Downey, for 30 minutes. And yep, just use the raise hand function if you have questions at the end. And now I'd like to pass it off to Matt to give a little introduction to the CSMB.
Awesome, thank you. So welcome everybody to our first Canadian Society of Molecular Biosciences virtual seminar. I'm Matt Berg, a postdoc and the trainee representative on the CSMB board. For those who don't know, I just wanted to give a little intro to the CSMB. So we're a professional association of active researchers in biochemistry, cell biology, molecular biology, and genetics. And there's many things that the society does, including participating in science advocacy and policy. We host an annual theme-oriented meeting. We give out awards to recognize the accomplishments of Canadian scientists, and we support the next generation of scientists. If you're interested in becoming a member, which includes many benefits, including discounted registration to our annual conferences, or if you want to renew your membership, you can sign up at the website here. So with that, I'll turn it back over to Kathy to introduce our speakers.
Thank you for that. So for our first speaker, we have Laura Orofiamma, and she is a PhD candidate at the Toronto Metropolitan University. Take it away, Laura.
Thank you so much. I'm just going to get a minute to share my screen. Okay. All right. Everyone can hear me okay? Perfect. So yes, my name is Laura Orofiamma, and I just wanted to say, first of all, a big thank you to the CSMB Virtual Seminar Series organizers for this opportunity for Canadian researchers and trainees like myself to talk about our work. So I am really excited to be here talking today about one of the broad questions of my research, which is what happens to cell physiology during a metabolically stressful event. So a metabolically stressful event could be considered an insufficiency in a key metabolite like glucose or oxygen. And in order for a cell to survive, it needs to be able to sense these insufficiencies and then adapt its means of energy production and also consumption. And so in all of our cells, we have a major metabolic energy sensor protein known as AMP-activated protein kinase, or AMPK. And AMPK is activated by a variety of insufficiency signals like low ATP or low glucose levels, among others. And once AMPK is active, it promotes energy-producing processes like glucose uptake and glycolysis, while also down-regulating energy-consuming processes like biosynthetic pathways, as well as the cell cycle. And one way that AMPK is able to regulate many of these diverse processes is by controlling what proteins are on the cell surface, or the cell surface proteome. And so AMPK has been shown to regulate the amount of glucose and fatty acid transporters on the cell surface during metabolically stressful events. And it does this by controlling their membrane traffic, or their endocytosis, recycling, and exocytosis back to the plasma membrane. And so we reason that there were probably other cell surface proteins that AMPK can regulate. So we performed a cell surface biotinylation assay and detected these purified proteins by mass spectrometry during AMPK activation with an allosteric activator, which we'll call A7. And we found that when AMPK was activated, we found quite a few interesting proteins that were depleted from the plasma membrane during AMPK activation. There were many that were involved in cell adhesion and migration, like beta-1 integrin and other related integrin receptors, as well as proteins involved in vesicle transport and endocytosis, like regulators of ARF-GTPase proteins, which not only have roles in integrin traffic, but also key roles in cell migration. And so we first wanted to explore how AMPK might regulate these integrins to control cell physiology. So we first looked at how much cell surface integrins are on the cell surface before and after AMPK activation. And we saw that AMPK activation causes a drastic reduction in the amount of cell surface integrin, but silencing AMPK does not produce that effect. And so given that we saw this at relatively short time periods, maybe about 5 minutes or so, and also some other confirmatory experiments, we reasoned that it's probably due to the increased internalization or endocytosis of these integrin receptors during AMPK activation. And so we then wondered what about AMPK activation controlling this process could impact cell migration? And so we analyzed non-directional or random cell migration during AMPK activation, and we saw that a variety of methods of AMPK activation were able to significantly reduce their non-directional cell migration, which of course is a very energy-intensive process. So it makes sense that AMPK would downregulate this. And we wanted to explore how this was working. So the connection between integrin endocytosis and cell migration. And what we know about this is that integrins are coordinated with their endocytosis from the posterior end of the cell and internalizing these integrins from, let's say, focal adhesions and either degrading them or recycling them back to the leading edge of the cell during cell migration. And so the mechanism by which these integrins are endocytosed from the plasma membrane is by clathrin-mediated endocytosis. And so we wondered if AMPK might be able to regulate this process to control cell surface beta-1 integrin and cell migration. So very briefly, I love clathrin endocytosis, so I have to give a big shout out to this process. It involves the protein clathrin, which is a really interesting triskelion-shaped protein with heavy and light chains. And it self-assembles into this cage-like lattice on the plasma membrane, and inside of it are containing the cargo proteins from the cell surface, and it can carry them into the cell. And so during the first stages, we have like cargo selection and clathrin recruitment that are mediated by a variety of clathrin-associated proteins. And if this process actually makes it to maturity, we get the recruitment of other clathrin-associated proteins like the fission factor dynamin to cut the clathrin-coated pit from the membrane and form an internalized clathrin-coated vesicle. And so AMPK has a number of potential different components that it could regulate in this process. And so we wondered if AMPK can broadly control endocytosis. We wondered what signals AMPK might be able to use to regulate integrin endocytosis specifically, and how this might impact cell physiology in terms of cell migration. So we first used retinal pigment epithelial cells that stably express clathrin light chain fused to GFP, and we treated them with the AMPK activator A7. And we saw a global reduction in clathrin intensity. And this tells us that the clathrin-coated pits are actually smaller during AMPK activation. So I like to think of it as nutrient-replete conditions giving the ability for cells to form the Hummer of clathrin-coated pits. They're quite large and they require many resources to do the job. Okay, but AMPK activation causes these clathrin-coated pits to be smaller and perhaps use resources more efficiently, kind of like a smart car. So we may be wondering, okay, if AMPK activation creates these smaller clathrin-coated pits, how are we still getting more integrin endocytosis? So we thought we might look at how much integrin is in these individual clathrin-coated pits. And what we saw was that during AMPK activation, there is significantly more integrin in these clathrin-coated pits compared to the basal condition. So it's kind of like trying to stuff a bunch of people into a smart car, okay? It's definitely a more efficient way of moving people, but I would say maybe not so comfortable. Okay, so then we asked, all right, AMPK might control integrin recruitment to these clathrin-coated pits, but how is it doing this? So in order for cargo-like integrins to be recruited to clathrin-coated pits, we need an adapter protein that's able to bind both integrin and clathrin. And here we focused on one of the integrin adapter proteins, DAB2. And we looked at how much DAB2 was in clathrin-coated pits during AMPK activation and saw a significant enrichment of DAB2 in these pits while AMPK was activated. And so we have this model now where AMPK is increasing DAB2 and integrin recruitment to regulate endocytosis and cell migration. But we didn't think that the interaction between AMPK and DAB2 was direct. And so we thought back to our mass spectrometry screen and all of these regulators of ARF-GTPase that popped up. And we thought, hey, probably an ARF-GTPase is regulated in this process. So we chose to focus on ARF6 for two reasons. The first is that it's the GTPase most associated with the plasma membrane and also has significant roles in integrin traffic and cell migration. And so with the help of our very wonderful collaborators from the Gingras lab, we wanted to determine if ARF6 had any interactors in this process. And so they performed for us a beautiful bioID screen for ARF6, as well as two mutants, one that's GTP locked and one that's GDP locked. And not only did we see this really great interaction between ARF6 and integrins, as well as clathrin-associated proteins, including clathrin itself, but also a GTP-dependent interaction between ARF6 and DAB2. And so from this, we reasoned ARF6 might be in clathrin-coated pits, and ARF6 might be able to control the way that DAB2 is recruited to clathrin-coated pits. So to look at this a little bit more, we generated a doxycycline-inducible stable cell line with ARF6 fused to GFP, and we used turf microscopy to specifically visualize about 100 nanometers into the plasma membrane. And what we found was that ARF6 does localize to some, but not all, clathrin-coated pits, and this is inherent in the heterogeneous nature of clathrin-mediated endocytosis. We also confirmed that DAB2 localizes to some, but again, not all clathrin-coated pits. But are they in the same clathrin-coated pit? And to answer this, we used a systematic thresholding system to characterize clathrin-coated pits that are either positive or negative for ARF6. And we saw that DAB2 recruitment was significantly increased in clathrin-coated pits that contained ARF6 than those that do not. And so this suggested that ARF6 might be involved in DAB2 recruitment to clathrin-coated pits. But again, we didn't think that this interaction was direct, and we pulled out one of the specific ARF-GTPase regulators, ARF-GAP3, from our mass spectrometry screen. And I kind of looked at the sequence and noticed that there were two potential sites with pretty high sequence similarity to the AMPK substrate consensus sequence. And although I still have yet to determine which specific one it is and the potential role of the phosphorylation, we decided we do have evidence to show that ARF-GAP3 is able to regulate the localization of ARF6 in clathrin structures. So we see that with AMPK activation, there's significantly more ARF6 in these clathrin-coated pits. But when we silence ARF-GAP3, it impairs this ARF6 enrichment in response to AMPK. And so to kind of put all of these big ideas together and determine what the specific roles are of ARF-GAP3 and ARF6 in this process, we silenced either ARF-GAP3 or ARF6 and was able to measure DAB2 recruitment to these clathrin-coated pits. And we did see the expected increase in DAB2 in clathrin-coated pits after AMPK activation. However, this did not happen when we silenced ARF-GAP3 or ARF6. And so this provides pretty good evidence for suggesting that both of these ARF-GTPase associated proteins can regulate DAB2 recruitment. But what about does the signaling pathway impact cell surface beta-1 integrin, like I showed you in some of the previous slides in my talk? So we again did silencing of ARF-GAP3 and ARF6, but I'm just showing the ARF-GAP3 data here for some clarity. And we saw that AMPK activation was unable to reduce cell surface integrin when ARF-GAP3 or ARF6 was silenced. And so this gave us a pretty good indication that yes, AMPK does require ARF-GAP3 and ARF6 to enrich these clathrin-coated pits with DAB2 and integrin for some more efficient integrin endocytosis during some metabolically stressful conditions. And so this leaves us with my working model so far, which suggests that AMPK, once activated by nutrient insufficiencies or other triggers, can use the GTPase ARF6 and a novel GTPase regulator, ARF-GAP3 for ARF6, to localize DAB2 and enrich DAB2 in these clathrin-coated pits, and DAB2 acting as the endocytic adapter protein for proteins like integrin can affect cell migration, but also may affect other proteins and other receptors that may use DAB2 as an endocytic adapter protein in this process. So there's lots more to investigate here that I'm really excited about. So with that, I want to thank my lovely supervisor, Dr. Costin Antonescu, all of my fellow lab members. Thank you to our wonderful collaborators and our funding sources. Thank you again to the CSMB organizers for hosting the seminar series, and thank you all for listening to my talk today. Happy to take any questions. Thank you very much.
All right, so do we have any questions? Amazing talk, Laura.
Thank you.
I can ask a question if nobody else has any. So you have the hypothesis that there are potentially phosphosites on what is ARF-GAP3 that are regulating this. Is there any evidence in any phospho mass spec data sets that those sites are phosphorylated upon AMPK activation?
Yeah, that's a really great question. So there are actually really good yeast studies that have looked at the yeast form of AMPK, which is SNF1, and the yeast form of ARF-GAP3, which is GLO3. And what they found, if I can just go back to the slide for just a minute, what they found was that this GLO3 regulatory motif was in fact phosphorylated by the yeast form of AMPK. So there's really good homologous data for this. So we're just trying to confirm that now in mammalian cells. But yeah,
Cool.
That was really interesting. Thank you.
Super cool. And we'll go to Patrick Lejeune now.
Hello. Yeah,
Hello.
very good talk. So my question was, can the AMPK, do you know if it can impact non-clathrin regulated endocytosis? I know RF6 regulates caveoli endocytosis, so can that play a role in some of your system?
Absolutely. AMPK can regulate other types of endocytosis. There's also some evidence to suggest that AMPK can regulate some ARF6 independent endocytosis, which is really interesting. So we are trying to devolve between the differences on these other types of endocytosis and other proteins that are involved in cell migration that AMPK could regulate. But yeah, this is kind of the story that we've put together with the clathrin focus, but definitely much more room to expand.
Great, thanks.
Thank you.
And we have one more question from the chat from Claire. Fantastic talk. Can you explain your surface protein screen following AMPK activation a bit more? Aren't our ARAP1 and ASAP protein cytosolic?
Yeah, absolutely. So what this screen detects is sort of the membrane fraction and what proteins come in and out of this membrane fraction during the cell surface biotinylation. So ARAP1 and ASAP1 are mostly cytosolic, absolutely, but can exist sort of at a closer fraction to the plasma membrane, which is probably what we've been picking up, and can kind of dissociate and associate from that fraction. So again, they do interact with ARF6 as well, but we followed the ARF-GAP3 story because we thought that that was really novel and interesting. So thank you for that.
Great. I hope that answered your question, Claire. So in an effort to keep us on track time-wise, I'm just going to ask Laura if you can stop sharing your screen. And awesome.
Thank you so much.
Of course. All right, so our next speaker is Dr. Mike Downey, and he is an associate professor at the University of Ottawa. And today he'll be presenting his work, Systems Biology of Polyphosphate, and I'll let him take it away.
Okay, great. First, I just want to make sure that everybody can hear me okay. Yes? Okay, great. And you can see my little laser pointer here. Okay, fantastic. Okay, so I'm going to be talking about a molecule called polyphosphate today. Let me just make sure I got my timer set here. And I'm going to be telling you about kind of a weird way that we got into the polyphosphate field, and now how our interests have kind of expanded a little bit across a variety of different model systems. Okay, so you're going to see a little bit of mammalian cell stuff today. You'll see some yeast and some bacterial work as well. And so the work that I'm going to describe is somewhat high level. My goal here is to kind of give you a flavor of all the different things that we're doing in the lab. And so to start off, I want to acknowledge the people that have done and that are doing the work. And so I'm going to talk about a little bit of stuff from Liam McCarthy today, PhD candidate, Kanchi Bajal, PhD candidate in the lab, and Alex Denoncourt, a technician in the lab, as well as previous trainees, Amanda Bentley D'Souza, who's now at Yale, MSc students Charlotte Hullinier and Rowan Nasser, and a postdoc, Emma Bondi-Troni, who's now at Health Canada. And so my lab, we're interested in a variety of different processes, different things across the desk all the time, and we're interested in kind of diverse aspects of biology. A lot of what we focus on relates in one way or another to this idea of post-translational modifications. And so I like to think of post-translational modifications as something that can specify function and also generate diversity within the cell. And so if you take this very simple schematic here, you have a gene, and you can have various splicing events to lead to different mRNAs. Maybe you have some variable initiation of translation, but ultimately you're going to produce different protein species. It's the job then of post-translational modifications to add either chemical or small protein adducts to these polypeptides to generate this type of functional diversity that I was talked about. And so there's a lot of different post-translational modifications out there. Some of these maybe you know a lot about. Others, as a field, such as acetylation, we know a lot, but only about a very narrow substrate range. And so my lab actually studies one part of it, acetylation. If you're interested in acetylation biology, I'd love to chat sometime. And so a lot of the different types of modifications that we've been interested in over the years happen to be lysine-based modifications. And like post-translational modifications in general, there's a lot of different lysine-based modifications. Some of these are chemical-based, like acetylation or methylation, and some of these are protein-based, such as summoylation or ubiquitination. And so the interesting thing about lysine modifications is that there's a lot of crosstalk going on. And actually, we think that lysine-based modifications can even compete with one another. And so usually you don't think that a single lysine residue could be both acetylated and ubiquitinated at the same time. And so I always argue to people that, whether you're interested in a protein that's methylated or even just simply something that's turned over by the ubiquitin proteasome system, and, this is going to be a very large fraction of your proteome, then really anything else that's going on within this biological space is something that could impact your work. And so it's something that you should think about and be interested in. And so today I'm going to be telling you a little bit about something called polyphosphorylation, which we think may be a new lysine-based modification. Okay, so polyphosphorylation. Now I have to tell you about polyphosphate. So what is polyphosphate? Well, to us, polyphosphate is an interesting molecule. It's probably the most interesting molecule you've never heard of before. And so what are polyphosphate chains? Really, they're exactly what they sound like. So they're long chains of inorganic phosphates. So these can range from anywhere from, 3 residues is technically a polyphosphate, all the way up to thousands of residues. They're found in most cell types where people have looked to date. Plants might be an exception. We're not sure if they exist there. And they're joined by really high energy bonds. So these are the same types of high energy bonds that you might find in ATP as an example. And so in yeast, we know that these are made by an enzyme called VTC4. And so VTC4 sits on the vacuole membrane. So in yeast, the vacuole is analogous to the mammalian lysosome. And so it sits on this membrane in the context of a larger complex called the VTC complex. And it actually takes ATP, it essentially rips off that terminal gamma phosphate and adds it to a growing chain. And so these polyphosphate chains accumulate to really high levels in the vacuole. And when I say high levels, I mean it. So it's up to 10% of the dry weight of a yeast cell consists of polyphosphate. Okay, so as somebody who had studied yeast biology for 15 years before I even heard the phrase polyphosphate, this was really surprising to me. It's really interesting. And so I think this had sparked our interest in this field in large part. And so high concentration of polyphosphate in the vacuole, naively, we can think of the vacuole as kind of a bag of polyphosphate. It's thought that it can make its way to other cellular compartments, so the nucleus and maybe the mitochondria. How that happens is not really well understood. Is it somehow synthesized locally using different mechanism? Is it transported? Really, we don't know. You could imagine that perhaps it involves the action of some of these phosphatase enzymes called exopolyphosphatase enzymes. These can chew up polyphosphates from the end of the chain, or endopolyphosphatases that can cleave in the middle. So if you can make smaller chains, maybe this can impact export out of the vacuole. And so what's interesting in human cells, we know that there's polyphosphate. We think it's important, but we don't know the enzymes that make it, okay? Despite this, we know that across eukaryotic systems, and as you'll see, prokaryotic systems, polyphosphates play a variety of important roles. So some of these are maybe not that surprising. So phosphate metabolism, well, it's not that surprising that a long chain of phosphates is somehow integrated into phosphate control. Ion homeostasis, okay, well, it's a big negatively charged thing. Probably it's binding positive ions. But there's also a variety of roles in cell signaling, protein folding, and actually a pretty neat role in blood clotting, okay? So polyphosphate, very simple molecule, how is it exercising these diverse functions? And so you can imagine kind of a variety of mechanisms, and some of these are hypothesized, some of these have been demonstrated, and they're not necessarily mutually exclusive. And so it could act by chelating ions. I've mentioned this already. It can interact with proteins non-covalently. It can serve as a signaling role. And so this has been proposed to occur in neurons in the brain. And again, what I'm going to talk about today is this idea that it can covalently attach to proteins. And again, these different mechanisms maybe are not going to be mutually exclusive. So all this idea that polyphosphate can be added to proteins as a PTM comes from this study published in 2015 from a group in the UK, the CRD lab. And so what they found is that these chains can be non-enzymatically added to two different protein targets in yeast. And so you should go and check out this paper. I'm going to give you kind of the 4 things you need to know to understand the rest of the talk. And so first of all, when we started this work, only 2 known targets, both in yeast, NSR1 and TOP1. NSR1 is an mRNA binding protein. TOP1 is DNA topoisomerase. And these guys actually interact. And there's some evidence that modification with these chains can actually impact their action or their interaction and perhaps their subcellular localization. As I mentioned, it's non-enzymatic. So you make the chains, and then the thought is that it's added as a group to the protein target. In both of these protein targets, NSR1 and TOP1, it seems to be modified in areas which we're now referring to as PASC motifs or PASC domains. It stands for polyacidic, serine, and lysine rich. So lots of Ds and Es interspersed with serines and lysines. And so the interesting thing, of course, is that, normally we think of anything related to phosphorylation. What are you thinking of? You're thinking of serines, you're thinking of threonines, and you're thinking of perhaps tyrosines. And so in this case, the modified lysine seems to be, the modified residue seems to be lysine. And so they termed this modification lysine polyphosphorylation. Okay, and so when we read this paper, we wondered, what is the scope of lysine polyphosphorylation? Is this something that is a quirk of the biology of NSR1 and TOP1 in yeast? Or does this have the potential to be a global protein regulator? And this fits in with our interest in other lysine-based modifications, such as acetylation. And so what we decided to do first was say, well, are there any other proteins in the yeast proteome that look like NSR1 and TOP1 in terms of containing these PASC motifs. And while bioinformatics with two proteins, starting just with two proteins and looking at their sequences, it doesn't really get you all that far. So we essentially came up with a definition that looks good. And so people often ask me, oh, could this be an exact definition? It's like, well, no, it's not an exact definition. It's something I made up pretty much on the spot, okay? And so, but if you take this definition of a 20 amino acid sliding window, 75% of these residues that look like they're enriched in that PASC motif, we added at least one lysine, of course, because that's the proposed site of modification. You actually pull up to 7% of coding sequences in yeast. Okay, so up to 427 different proteins. So we're looking like there might be some room for some of these modifications occurring. And actually, it's interesting, if you look at kind of the biological processes associated with these different proteins, you can see that you're already seeing an enrichment for various processes related to RNA biology. And that's something that's going to come back again and again throughout the presentation. So then we ask, are any of these proteins going to be polyphosphorylated? Are they going to be modified by polyphosphate in the same way that was described for NSR1 and for TOP1? Well, often with post-translational modifications, you jump immediately to a mass spec-based approach. There's no defined mechanism to look at polyphosphorylation using mass spec yet. Obviously, it's something that we're interested in. For now, we diagnose polyphosphorylation using these so-called new-page gels. New-page is a denaturing bis-tris gel. Many people use these as a matter of routine in their lab without really understanding that they're not exactly the same as SDS page gels. And if that includes you, I can mention a few caveats associated with using these gels after the talk. And so notably, SDS page gels, and this will come up later as well, these do not resolve these polyphosphate-induced shifts, okay? And so this is our work reproducing the original study with TOP1. And so if you have TOP1 tagged with the TAP tag, and so this is just an epitope tag that we can detect using a Western blot, you can see that this is where the protein is expected to run. This is just based on molecular weight, but it only runs in this position in a strain that is missing VTC4. In the presence of VTC4, so an otherwise wild type strain, you can see that it runs with this very exaggerated molecular weight, okay? And so the reminder then that VTC4 is thought to make these chains, the actual addition is thought to be non-enzymatic. So great, we have a list of targets, we have a potential assay. How are we going to do this for these 400 different proteins? Well, what we did is we took advantage of a set of strains we have in the yeast community where each open reading frame is expressed as a fusion with the GFP epitope. So we're very fortunate if we want a GFP tag strain, we just go to the freezer. We have 6,000 different strains covering the entire yeast proteome. We can pull out a strain that is tagged at the genomic locus expressed as a fusion protein with GFP. So we can retrieve our 400 candidates that look pretty cool, and we can use some high throughput genetic techniques to generate two sets of strains. So the first of these is, it's kind of boring. It's just a control set, right? And so this is exactly what we're starting with. That's good. You need a control. The second is expressing these various candidates, but deleted for VTC4. Then we can simply go in, grab the same fusion in both a wild type and VTC4 delete background, so plus polyphosphate, minus polyphosphate, and make a paired protein extract and run them on these gels, okay? And so this turns from kind of a high throughput idea to, at best, kind of a medium throughput idea, but it's been something that's been quite successful for us. And so far, we've uncovered 27 novel targets. And so most of these are now published. There's a smattering of ones that we kind of continually come across in studying various other aspects of polyphosphate biology. And so you can see in this list, again, well, I think you have kind of diverse functions, but definitely you have things that are enriched in various aspects of RNA biology. That's not surprising because our initial group that we prioritized for screening were jackpotted for proteins belonging to this functional group. You can see as well that they are also, many of these proteins are conserved in human cells. Many of them are associated with diseases. And the ones that are in bold are human proteins that contain these PASC-like motifs. So some great opportunities for conservation here. Okay, So this is the current landscape of lysine polyphosphorylation in yeast. And so this is one of these kind of hairball diagrams that those of us in the systems biology world like to draw. And so, but in this case, I think it's actually pretty useful because it defines some areas of common functional overlap. And so in this diagram, each circle represents one of our modified proteins, including the two that were identified previously, TOP1 and NSR1. And a kind of a pink or salmon color line here represents a known physical interaction curated from the literature. Green represents a genetic interaction. So what we see here is that all these proteins seem to be largely connected in some way, which again is helpful for us because it can point us in various directions as to where to go in terms of follow-up studies. And I'm actually going to briefly mention various aspects of these along the presentation. So this is what the running these hits on these new page gels actually looks like. This is rerunning our hits or reassaying our hits. We didn't get lucky and find 6 in one go. But let me just take you through this so that we're all on the same page. And so what we're doing here, again, we're running on these denaturing gels. We have in each case plus VTC4 and then a strain that is missing VTC4 and expressing the fusion that's indicated along the top here. And then we're detecting the fusion with an antibody against GFP. Okay, so here we're not so interested in GFP for its fluorescent properties. We're simply using GFP because it's a great epitope to detect in Western blotting. And so you see these very dramatic shifts for all these different proteins, very quantitative. One of the things we did notice almost right away, though, is that the degree of shift actually differs depending on the target that we're talking about. Okay, and what we found is that this scales very nicely with the number of lysines that are located within the PASC motifs. So this is great because this is what we would expect from this type of a modification. It's also interesting because it suggests that some of these targets might have multiple chains attached. Okay, and so this is similar to how we think of many other chain type modifications, such as pyrrolation or ubiquitination. Okay, and so the idea here is that function could be determined by the number of chains that are attached. And so we saw no such correlation with the number of acidic residues or the number of serines within the PASC motif. So this is something which is specific to these lysine residues. So as further evidence, these PASC motifs are important. What you can do is you can go into the individual targets and you can delete the PASC motifs and see if you're losing that shift. And we are fortunate that a number of our targets, and actually most of our targets, seem to have these PASC motifs towards the N or the C terminus, often in intrinsically disordered regions, okay? So those on the C terminus are very easy to delete genomically in yeast. We simply replace the C terminus with the GFP tag within the genome. And so I'll take you specifically through the work we did with RPA34. RPA34 is a subunit of RNA pol1. And so this is the shift that we're used to seeing with this target. So nice, very quantitative shift. When we delete the PASC motifs, you can see that the proteins are running lower on the gel. This makes sense. We're deleting a chunk of the protein, but you see no contribution of VTC4. Okay, so this indicates that PASC motif is required for modification. Now, for RPA34, we've actually gone in one step further. We've assayed the contribution of these various lysine residues. And so what we can do is take this PASC motif and mutate each lysine residue to an arginine. So similar charge, but different chemistry. And so if this is the wild type RPA34, and in this case, it's running a little bit more of a heterogeneous smear, and we think this is because it's overexpressed in this assay. When we mutate K to R, you see that you lose this shift, and this is relevant to the VTC delete controls, where that shift is expected to be completely collapsed. What's more is that these PASC motifs seem to be portable. So we can take a strain that is lacking the RPA34 PASC motif, and we can substitute it with a PASC motif from another protein, and indeed we restore this shift. So all of that information that is required for that so-called non-enzymatic reaction, this seems to be contained within that PASC motif or domain, okay? So the other aspect is that I mentioned that polyphosphorylation, the idea is that it's a non-enzymatic modification. And this can present some real challenges when trying to get at in vivo function. Why? Because the idea is that when you have a target and you make a lysate, if there's polyphosphate in that lysate, it's going to modify it in vitro. So this is definitely a technical hurdle that the entire field has struggled to overcome. But it does allow us to do a really important rescue experiment. And so we can start with an extract that is deleted for VTC4, and we can add back polyphosphate that is made synthetically. When we do this, we can restore this shift to a strain that is lacking VTC4. In this case, we're doing this for this target RTS1, which is a subunit of the PP2A complex, and we're detecting RTS1 with an antibody against the native protein. So this works in extracts. It also works with purified protein. So you can take a fragment of RTS1 that contains the past motifs, add in polyphosphate to get a shift, and we don't get a shift with GST alone. So what about function? Well, we're as you can imagine, diving in and looking at the function on individual proteins is a long process. We're still going after a number of these targets. But what I want to talk about today is something that we notice when we look at this group as a whole. And so when I showed that big network diagram, you probably saw this area that's enriched for proteins that localize to the nucleolus. And in fact, all of these proteins are involved in various aspects of ribosome biogenesis. And so based on this, we've been able to tease out a new phenotype for VTC4 delete cells in polysome assembly. So polysome assembly measures the ability of ribosomes to associate with RNA. Okay, and so in VTC4 delete cells, you can see that the assembly of these polysomes is greatly reduced. And so the idea then is that this may be due to polyphosphorylation of one or more of these targets. It's also possible that this phenotype is unrelated to polyphosphorylation and is due to some other aspect of polyphosphate biology. Importantly, as we delve into more diverse aspects of polyphosphate biology, we consider this possibility to be equally interesting. Okay, what about other species? And so everybody always wants to know about human cells. Well, there's a problem here. And so one of the problems is we don't know the enzymes that make polyphosphate in human cells. It's somewhat of a mystery. And at least our initial screen relied on this idea that we knew the identity of VTC4. Also, poly-P levels are quite low in most human cell lines. And so too low to introduce these very quantitative and probably exaggerated shifts that we see in our yeast assay. And so what we did is we took a step back and we said, well, if there were to be a condition where there was a lot of polyphosphate, perhaps locally within an area of the cell or under some as yet unknown condition, would that polyphosphate be able to modify human proteins that contain some of these past motifs? And so what we did is we stole an enzyme from bacteria that can make polyphosphate independently of the vacuole, transfected this into HEC293 cells. And what you see here is that this allows the production of high amounts of polyphosphate. Okay, so this is just showing the polyphosphate accumulation. This is various size standards of polyphosphate. And basically, this is a page separation of polyphosphate. And the polyphosphate chains in this case are actually detected using DAPI. Okay, maybe not surprising that polyphosphate can be detected with DAPI, knowing that it also detects other negatively charged molecules such as RNA and DNA. And so you can see in the whole cell lysate, we're detecting polyphosphate. And also when we fractionate the cell, you can see that it's really accumulating throughout the cell. Okay, so this works. Okay, so when we do this, then we can start to see polyphosphorylation of different proteins. Okay, so EIF5B, UPF3B, MESD, and showing here DEC. Okay, so DEC is a chromatin protein involved in various aspects of transcriptional control. Okay, and so we like to think that what we're seeing here in this E. coli PPK system, where we're artificially increasing the amount of polyphosphate, this is equivalent to overexpressing your enzyme of interest within a cell, okay? So if you can't delete your protein, but you can overexpress it, this is really kind of what we're doing in this situation. So we would like to think that what we're observing here may be an exaggeration of what might be happening perhaps at a more selective level in vivo. And so what's really neat is that there's also over 1,000 PAS-containing proteins in human cells. These localize to diverse compartments, and so we think there's a lot of room for regulation in this sense, okay? What's more, we think that polyphosphate might be doing interesting things. And so for DEC, for example, we've noticed when we analyze, in this case, on SDS page gels, remember, no shifts in this case. When we express PPK, you can see that it changes the localization. So you're basically moving DEC out of the nucleus into the cytoplasm and organelle fraction. Okay, so polyphosphate seems to be doing important things. What's more, and this is really just kind of an advertisement for this paper, is we've actually used transcriptomics and proteomics to look at diverse pathways which are misregulated when we express PPK. Okay, and so as an example, we see upregulation of ERK signaling and some aspects of TOR signaling when polyphosphate is being overproduced within these cells. Okay, so using this system allows us to get at the types of proteins that could be modified and also to identify functional areas where these proteins might be playing important roles. We've also gone the other direction. So we found that bacterial proteins can actually be polyphosphorylated as well. And so we discovered these candidates using a screen very much similar to what we did in yeast, okay, by taking a collection of strains and really kind of screening through them very systematically. And bacteria may be a really ideal system to study this phenomenon of polyphosphorylation. And the reason for that is if you think about yeast as an example, this is our best characterized system in terms of polyphosphate biology. We see that most of that polyphosphate is locked away in the vacuole. So if you've got a nuclear target, you want to be thinking about function. First, you have to start to answer these questions about, is polyphosphate getting to the nucleus and how does that occur to get at function? Now, we want to answer those questions, but it's going to take some time. In bacteria, they make polyphosphate, and at least naively, it's not associated with any particular organelle. And they make polyphosphate specifically when they're stressed out. And so there's already kind of phenotypes you can begin to imagine. So you can start to think about the role of polyphosphate in various stress responses related to bacterial physiology. Okay, so previously, polyphosphate has been implicated as an important player in bacterial motility, infection, and various aspects of survival during stress. And for this reason, it's been proposed actually as a novel drug target. And so one example of kind of the phenotypic work that we've been doing in bacteria is to look at the role of PPK. This is this enzyme that makes polyphosphate in bacteria in terms of antibiotic resistance. And so this is work from Conchi in a strain background that is resistant to polymyxin. And in this case, you can see that mutation of PPK reverses that resistance to this drug. And so the idea here is that if you can inhibit PPK using a drug, then perhaps you can start to use lower levels of antibiotics, and perhaps some of these polyphosphorylated proteins are involved in this type of response. Okay, so this is where we're going in the bacterial world. Now, it's interesting, this idea of drugging PPK is not just some sort of a pipe dream. Recently, there was a study published suggesting that a drug called mesalamine has been used since the 80s to treat Crohn's and colitis. It actually functions as a PPK inhibitor. Okay, so there's some precedent that this might actually work. So I just want to say in the last few minutes, a few words about this concept of polyphosphorylation and how it might be regulated. Okay, so let's think about this for a moment. If you have a modification, let's say you have a modification like acetylation. If you think about regulation, usually you think about regulating the enzyme doing the modifying. So you have an acetyltransferase, let's turn it on or off, degrade it, move it somewhere, these types of things. Well, there's no enzyme here, it's non-enzymatic. So how do you regulate this type of a process? Well, there's a variety of different things that we can think about. First of all, when you have these chains attached to the protein, we think that there might be a few phosphatases that actually exist, such as PPX1 and DDP1 in yeast that can directly remove these chains. But also we see big potential for regulating the modification based on the production of the molecule itself, okay? So just like acetylation, for example, if you want to have protein acetylation, you need to regulate the levels of acetyl-CoA. If you want to regulate polyphosphorylation, you have to regulate the levels of polyphosphate. And it's through this type of logic that we become very interested in more fundamental aspects of polyphosphate biology, even beyond and distinct from polyphosphorylation. So just to mention a few ideas that we've been following, One interesting observation is that we see that, there's these phosphatases. One of them is called PPN1, the other is called PPN2, which have been shown to degrade polyphosphate within the vacuole. Well, we actually see that PPN1 is a target of polyphosphorylation. So we think there's some interesting room here for negative or positive feedback in terms of PPN1 function. We've also been active in looking at mechanisms in regulating polyphosphate synthesis. And so in particular, we saw previously in the previous presentation mention of AP2. Well, the AP3 complex is a protein transport complex that we found is important for regulating VTC localization to the vacuolar membrane. If you begin to mutate these different subunits, you see loss of polyphosphorylation, in this case stained with something called toluidine blue. This complex is conserved, and because it's conserved, we think that following this biology might actually lead us to that human protein, that unknown human protein that makes polyphosphate. And finally here, we've done a screen for protein kinases that have decreases in polyphosphate levels. We're really interested in this conserved protein kinase called SCY1. This is an SR protein kinase originally identified in yeast as playing a role in splicing control. It's also involved in mRNA export, stress granule formation, and ion homeostasis. We think it'd be really neat to be able to connect polyphosphate and/or polyphosphorylation to any of these different processes. And so this slide serves as an example of how starting in the area of polyphosphorylation has led us to be more interested in these basic mechanisms of polyphosphate metabolism. And so in summary, what have we learned so far? So I focused on this today. Polyphosphorylation could be a common post-translational modification. And again, I've talked about how this initial interest has led us into more diverse aspects of polyphosphate biology in a variety of different model systems. And so we started as a yeast lab, and then we've begun to do more work in mammalian cells and more recently in bacteria. I mentioned the idea of the AP3 complex. We're really interested in delving into various aspects of bacterial genetics related to PPK. And something I didn't talk about today is this concept of localization. So it seems that it's actually important to regulate polyphosphate localization within the cell. If polyphosphate ends up in the wrong cellular compartment, cells don't like it and they seem to kick off a stress response. And actually, as it turns out, I'm going to be talking about this next week in the proteostasis seminar series. And so if you're interested, please attend that seminar. So again, just thanking the people in my lab. So the current lab, we're actually interested in recruiting new students. If you've seen anything that you're interested in, past trainees, fantastic collaborators. And so we've moved into a lot of different areas where we had no expertise, and we've had great help along the way, particularly in the areas of the bacterial side of things from Mike Gray and Stephen Trent. Funding agencies. So we're really fortunate to have our polyphosphate work funded by CIHR. And so usually we think of CIHR as being very health oriented. Obviously the work with polyphosphate is very discovery based. A lot of it is very descriptive and phenomenological, but I'm happy to see that reviewers have really kind of taken to this and understood the importance and the potential of this. And of course, I have to end by advertising a meeting that we're hosting. So this is the 66th annual meeting for CSMB. It's in Ottawa, May 30th to June 2nd. The topic is metabolic regulation of cell signaling, but it's really designed to be broad so that anybody involved in the CSMB community can attend, enjoy the meeting, and be considered for a talk. And so if your research doesn't fit perfectly into any of the themes, it doesn't matter, you're still going to be considered for a talk, okay? So we have a great keynote speaker, Marcus Rausser from Charité Berlin. We have trainee development workshops during the first day. I think you'll see the pricing for both the conference and the housing is very affordable. We have a super cool banquet at the Canadian Nature Museum, okay? So if you're at this 2017 meeting, it's the same venue. It's a really great environment for an end of conference banquet. 75% of all talks are going to be picked from the abstract. So this is a great opportunity to showcase your work in front of your colleagues. So please register for the meeting, register your team. Early registration closes on March 15th. So check out the website and let me know if you have any questions. Thank you.
Excellent. Thank you so much, Dr. Downey. That was a great talk. And we actually already have a question in the chat. So by Claire, you mentioned briefly about competition for lysine as potentially a flavor of regulation in the cell. Do you have any evidence of whether lysines can be simultaneously polyphosphorylated and ubiquitinated or sumolated, etc.? I'm thinking in the context of regulating something like protein degradation, for example.
Yeah, it's definitely a possibility. We don't have any defined examples of that, but you could easily imagine that polyphosphorylation might prevent ubiquitination that would then stabilize a protein. And so that's kind of first stop for any of the candidates that we've been interested in following. It's looking at the stability of the protein using things like cyclohexamide chase assays to look at the stability of the protein over time. And so that's something that we're really interested in looking at. Also, the idea of competition with other lysine-based modifications, such as acetylation. What's interesting is that, well, there's been no method developed to look at lysine polyphosphorylation via mass spectrometry. You can look at other modifications. So if you can find a site of, say, acetylation that changes the amount of acetylation in a VTC4 mutant, maybe that's a switch that's actually regulated by polyphosphorylation. And so this might be an indirect way to begin to localize sites using mass spectrometry. So great question.
Thank you. Any other questions?
Yeah, I have a couple depending on time. The first one is, so the mammalian stuff's really cool with the localization. You have the whole GFP collection in yeast. Have you used that to look at changes in localization in the VTC4 delete strain?
So it's definitely something that's on the list, right? And that would be a really kind of a great way to, like, if you're looking at, if you have a set of targets and you want to find something to follow up that's going to have a tractable phenotype, then that's something that we're definitely interested in doing. We've done that for most of our hits. We don't see any big giant grand changes, but even the changes that were described originally for, say, NSR1 and TOP1, they're actually quite subtle. So moving in and out of the nucleolus as opposed to the nucleus, things like this. And so I think what we'd want to do is kind of get somebody dedicated experience in microscopy who's interested in looking at that a little bit more closely. But that's a great idea. And it's a great way to prioritize these targets. And so, like any other post-translational modification or, even a non-covalent interaction of something, when you're picking things and pathways to follow up, you want to have something like a clear assay that allows you to kind of prioritize your targets. And so things like localization is something that is a great idea.
Totally. Sounds good.
We have time for one more. And I'm not seeing anyone else. Matt, if you have another question.
Yeah, we're at 10 o'clock, so I can keep it. I'm interested in the mass spec angle of this, but we can talk later, Mike.
Sure.
All righty.
All right. Thanks, everyone.
Thank you, Dr. Downey. Thank you, everyone, for attending today. And just briefly, as everyone is heading out, just a reminder that our second session is on March 14th. Feel free to join us again at the same time, and we look forward to seeing you all there. Feel free to stick around if you have any questions for the speakers. Alrighty.
That was awesome. Thank you both. I think that was a really great first seminar. You gave great talks.
Thank you so much again for hosting this. It's a really great opportunity for trainees as well. So thank you for thinking about that.
Yeah, it's awesome.
Okay.
All right. Take care, everybody. Nice to see all of you.
Okay. That's it.
Yeah, sounds good. Okay. I'll talk to you later.
Sounds good.
Okay. See you.
Bye-bye.